Molecular cloning is the technique of generating identical copies of a specific DNA sequence. This process consists of several critical steps. In this animation, we will break down each step, starting with the isolation of DNA from the target cell. We begin by taking the target cell in a tube. Next, we add a lysis buffer containing SDS and EDTA. This breaks open the cell membrane and nuclear envelope, releasing DNA, proteins, and other components. Next, we add protease enzymes to digest proteins. Then, using phenol chloroform extraction, proteins move to the organic layer, while DNA stays in the aqueous layer. Finally, we add cold ethanol to precipitate the DNA. DNA appears as a stringy mass. The second step of the cloning process is the extraction of the gene of interest. From the entire genomic DNA, we only want one specific gene, the one we plan to clone. To isolate it, we can use restriction enzymes, like molecular scissors, to cut the DNA around the gene. Or, if we just know the sequence, we can use PCR to amplify it. PCR lets us make millions of copies of that one gene segment from the whole genome. In both cases, we end up with a clean fragment of our target gene, which we'll use in the next step, insertion into a vector. A vector is a DNA molecule that carries our gene into a host cell. The most common type is a plasmid, a small, circular piece of DNA. We prepare the plasmid by cutting it open using the same restriction enzyme used to cut our gene. This ensures the ends are compatible. We then mix the plasmid and gene of interest together and add an enzyme called DNA ligase. This enzyme acts like glue, sealing the gene into the plasmid. The result is a recombinant plasmid, a circular DNA molecule now carrying our gene of interest. In the fourth step of molecular cloning, we introduce our recombinant plasmid into a host cell, a process called transformation. The most common host is a bacterial cell like E. coli. To make the cell take up the plasmid, we can use methods like heat shock or electroporation. In the heat shock method, we first chill the cells on ice with the plasmid, then briefly expose them to 42 degrees Celsius. This sudden temperature change opens pores in the membrane, letting the plasmid enter. After exposing the cells to 42 degrees Celsius, they are immediately placed back on ice. This sudden cooling helps the membrane reseal and stabilizes the cells after the heat shock. In electroporation, a short electric pulse is applied to the cells, which creates temporary pores in the membrane, allowing the plasmid to pass through. Once inside, the plasmid replicates along with the bacterial DNA, allowing our gene of interest to multiply. The next step is selection and screening. After transformation, not all bacteria will have taken up the recombinant plasmid. To identify the ones that did, we use a selection method. The most common technique is antibiotic selection. The plasmid we inserted carries a gene that makes the host cell resistant to a specific antibiotic, for example, ampicillin. We plate the transformed cells on agar that contains ampicillin. Only the bacteria that successfully took up the plasmid will survive and form colonies. The rest will die. To confirm this, we also plate the same cells on agar without antibiotic as a control, where all bacteria grow. This comparison shows that only the resistant bacteria survive on ampicillin plates. Another technique we use to select the recombinant plasmids is blue-white screening. The plasmid carries the ligase gene, which produces a blue color when active. If our DNA insert is successfully integrated, it disrupts this gene, and the colony appears white. So, white colonies contain recombinant plasmids, while blue ones do not. Other techniques involve colony PCR and fluorescence markers.
Please like, share and subscribe and turn the bell icon for more videos.